So it is my pleasure to welcome Dr. Juliana Rangel. And I'll say a few words just about Francisco Rangel's background. Um, Born in Colombia, South America, Juliana obtained a BS in Ecology, Behavior, and Evolution in 2004 from the University of California at San Diego. In 2010, she obtained a PhD in Neurobiology and Behavior from Cornell University in Ithaca, New York. She was an NSF postdoctoral research fellow from 2010 to 2013 at North Carolina State University. And in January 2013, Juliana became assistant professor of apiculture in the Department of Entomology at Texas A&M University. She was promoted to the rank of associate professor with tenure in 2018 and professor in 2023. Her research program focuses on the biological and environmental factors that affect reproductive quality of honeybees, the behavioral ecology and population genetics of feral honeybee colonies, and the quality and diversity of honeybee nutrition in a changing landscape. She is an active member of the Texas Beekeepers Association and has been invited to speak at dozens of scientific conferences and beekeeping association meetings across the United States and internationally, as our president Jan mentioned, having heard her in Calgary. She teaches the courses Honeybee Biology, Introduction to Beekeeping, and Professional Grant and Contract Writing. Since 2014, she has been the coach of TAMU's undergraduate and graduate teams of entomology games at the branch of national games of the Entomological Association Society of America, earning first and second place nationally for 4 years in a row. She was the 2023-2024 president of ESA's Southwestern branch and is the past elected chair of the National ESA's Diversity and Inclusion Committee. She previously served as the elected chair of her department's faculty advisory committee and has been part of several committees at the departmental, college, and university levels. And most recently, she received the 2024 Association of Former Students Award for Teaching Excellence, the 2023 Vice Chancellor's Award for Excellence in Teaching for tenure track faculty, and the 2023 Distinguished Achievement Award in Teaching from the Southwestern branch of the ESA. In 2021, she received the James I. Hamilton Memorial Award, which was established by the Eastern Apiculture Society of North America to recognize research excellence in apiculture. She's also received the 2020 John G. Thomas Award for Meritorious Service from the Texas Beekeepers Association for her contributions to the apiculture industry in the state. She received the 2019 Dean's Award for Excellence in Diversity and the 2016 Dean's Award for Excellence in Early Career Research from TAMU's College of Agriculture and Life Sciences. She also received the 2018 Astounding Achievement in Mentoring Award from the Entomological Graduate Student Association. She was the 2014 president and 2013 vice president of American Association of Professional Apiculturists. And obviously, this list of sort of honors, awards, positions, and also the emphasis on the teaching, I think that really comes through the impact of Juliana Rangel's work as a teacher. And I'm just delighted to invite her to present to us. And when we were talking about the presentation today, she sent me a list of possible topics. And I jokingly said the list was so great, we should just schedule her every month for a year, because they were such wonderful topics. Today, she's going to be speaking with us about queen management essentials. And with that, I'll turn it over to you. Thank you very much.
Thank you, Ian. I think when they asked me for a bio, I wasn't expecting it to be read out loud. I thought maybe it was just to place it on a Facebook page or something. So that was long, and I should probably cut it for when someone introduces me. I didn't mean to take so much time for you repeating everything I said. I'm sorry.
No
So
problem. It's my pleasure.
no,
It's
I'm
a
going
long
to
a long list of activities. So
okay, so can you see the screen or like because I have two screens, so yeah, you can see the main screen, correct?
we see at-home beekeeping webinar.
Okay, good. So before I move on, I just want to mention that I am part of this amazing program that started during the COVID pandemic. We used to be called Stay at Home Beekeeping Webinar Series when we were in lockdown and people couldn't get out of their house. But now it was so popular, people wanted some programming for beekeeping and they couldn't get together and like we normally do in person. So we did this online series of presentations and it became just kind of a staple now. So we changed the name to At Home Beekeeping Webinar. It's hosted by the Cooperative Extension Office in Alabama. And so it's kind of a weird named Lawrence County Extension out of pretty much a liaison with Auburn University in Alabama. The name of or the institutions that are part of this group the number has gone up. So it used to be maybe like 6 or 7 of us, and now we have groups from the Mid-Atlantic region, sometimes invited guests from the West Coast. So it's not just the Southwest or Southeast anymore. So it's free the last Tuesday of the month at 6:30 p.m. Central Time, and it's presented live both on Facebook and Zoom. So all you have to do is just Google "at home beekeeping webinar" and it's gonna show up the ways to link to Zoom or Facebook. You can watch it live, as I said. Otherwise, you can watch it at your leisure up to 2 weeks after it's presented live. The good thing about seeing it live is that there are moderators on both platforms, Facebook and Zoom, and so you can ask questions of the speaker and they will answer pretty much every question before the event ends. That's an advantage, but then the other advantage, if you're not watching it live, is that you can just watch it whenever you want. So the next presentation is next week by Melanie Kirby from the Institute of American Indian Arts in New Mexico, building comb between cultures and communities. And then on June 24th is my former PhD student, Liz Walsh, and I think I'm going to speak a little bit about her work when she was a PhD student here at Texas A&M. She's now a research entomologist at the USDA Bee Lab in Baton Rouge, Louisiana, talking about detecting viruses in a colony. If you go to their website, it's going to show who's coming up next. So normally we have about 4 presentations lined up for the next 4 months, and the only month that we don't have talks is the Christmas break. So I highly encourage you to watch these as a team or a club or just independently on Facebook. It's a really great way to learn what people, especially seasoned scientists, are doing in various organizations, institutions across the country. So today's talk, as Ian mentioned, he decided that I should speak about queen management essentials, and as he mentioned, one of my favorite topics of research is the factors that affect the reproductive quality of honeybee queens and drones. So there are several factors that affect queen health. And this is not an exhaustive list, but it's one that encompasses most of the issues that are affecting the health of our queens nowadays, including poor nutrition, parasites, in particular the varroa mite, pathogens, pesticide exposure, poor genetics, poor mating conditions, the age and vigor of the queens and drones that she mates with, climate change, habitat fragmentation, and unfortunately our own fault as beekeepers, poor beekeeping practices. And so I'm basically, for today's talk, I'm going to go over all of these particular aspects of what makes a good queen, or how a queen can basically fail due to these factors, and how as beekeepers we can play detective to manage these issues and maintain vigorous queens. So I'm going to start from the genetics portion of queen quality. So when we're queens, we have several things are going on in terms of genetics. We have the genetic strain of the queen. That means her race, where she's coming from, be it specific line like Varroa sensitive hygiene, Russian, Minnesota hygienic, or if you want to call it subspecies, either Ligustica, so Italian or Caucasian, or even Africanized, right? So that's one of the considerations. The other one is the genetic diversity of her parents. So how well bred her parents were, so that she has a lot of genetic potential to then impart onto her progeny. The type of breeding paradigm that she comes from, whether she comes from a completely open mated, naturally mated, non-bred program, or one that has been bred over generations to attain specific genetic traits that the beekeeper wants. And in particular, one that is very important is the degree of inbreeding, meaning the degree of how little genetic diversity there might be between her and the drones that she mates with. So we probably know, as beekeepers, you've learned about the fact that genetic diversity avoids inbreeding problems. It also increases specialization in the colony. It increases the potential for a colony to overcome disease, because there may be some specific genetic lines that can help improve the chances of these specific genetic lines being good at getting rid of certain pathogens. But the other extreme is inbreeding, of course. And so inbreeding causes the production of what we call diploid drones. And so you may have heard about diploid drones in the past and how they're not viable, but you may not know where those come from. So to learn about diploid drones, you need to know about the sex determining system in honeybees. So you've heard in the past that honeybees are haplodiploid, meaning that drones are haploid. They only have one set of chromosomes, 16 chromosomes in particular, and that females are diploid. They have two sets of chromosomes, so total of 32. Half come from the mom and half come from the dad. But the situation is actually a little bit trickier than that. Honeybee sex is actually determined by one gene called the CSD, or complementary sex determining gene. That has actually, I should change this number because I learned recently that in other countries, there are actually way more alleles than this. And in Australia, they found that there are more than 60 different alleles or variants or flavors. So this will bring us back to our biology and genetics 101 class in high school, where we have a gene that if you're a diploid organism, you get one copy of an allele from your dad, one from your mom, and basically you have a specific type of flavor or variant at that gene. And in this particular case is this sex determining gene. So if an egg is unfertilized by the queen, there's no sperm that's going in, then that individual only has one allele, because there's only one set of chromosomes at that sex determining locus, and therefore the bee develops into a drone, like we've learned all along. But the situation is specifically special or different if the egg is fertilized, because then the individual has two alleles at that sex determining locus. And so there are two possibilities: that the alleles are either the same or different. So if the alleles are different, which is what we want, then we call that in genetics heterozygous. And so the bee develops as a diploid female, which is the vast majority of individuals in the colony, especially workers. But in the unlikely event that those alleles are the same, therefore that individual is homozygous at that sex determining locus, that bee develops as a diploid drone. So let me explain a little further. Let's say that we have a queen, and as we mentioned before, queens are diploid, so they have two flavors or alleles at that locus, and we can either call it blue and yellow, or we can call it A and B, etc. And drones, because they're haploid, they only have one flavor of that allele, of that gene, so one allele, and they can be anywhere from A through Z, or however many alleles there are in that particular location at the sex determining locus. In this very simple scenario, there's basically four different ones. So A, B, C, and D are those alleles for the drones. and the queen is A and B. So by just random mating, that queen mates with a drone that has allele A, another drone that has allele B, C, another one with B, and one with D. If she shares any of her two alleles with any of the alleles of these drones, then there is at least 50% chance that her progeny are going to be homozygous at the sex-determining locus. So with A times A is going to make a homozygous AA, or AA or double A. In this particular case, the other half is going to be only AB. So this individual is heterozygous. That's going to turn into a diploid female. But these individuals that you see highlighted in yellow are going to be homozygous at the sex-determining locus, and therefore they're going to be considered diploid drones. So it's all basically random mating and how many times you're going to be drawing those dice and how many times you're going to end up getting pairs of the alleles. And so the higher the number of alleles available in the population, the less likely that a queen that has these two alleles, A and B, is going to mate with drones that share either A or B. What happens when the queen is highly inbred is that she is very likely to then start mating with drones that are of a genetic line that is related genetically to hers, maybe over several generations, and thus she's going to end up with these situations where you have homozygosity at that sex-determining locus. So it's random mating, but because she's mating with drones that may be related to her, then you increase that inbreeding potential, which creates the problem of diploid droids. So inbreeding causes a high chance of diploid bees having the same alleles at the sex-determining locus, and those diploid drones are not viable, and workers then remove them before emerging. So too many diploid drones cause what beekeepers call the shot root pattern, whereby all else being normal, there are no signs of disease, there's no signs of anything else going on. You may find something like this where you, instead of having a very even pattern of egg laying, you have a lot of open cells of what should be sealed cells. And it is because the workers remove those inviable drones from the cells before they even emerge as adults. They get cannibalized by the workers, likely because they smell differently than normal workers before they emerge. So that's a big problem, because then this reduces the productivity of the colony since there are no workers being produced. There was one study that showed that if they allowed scientifically or experimentally to for these drones to emerge from their cells, which they normally won't, they actually had many malformities in their adult body, including fewer wing hooks compared to haploid drones. And so that doesn't help with, these hooks are called hamuli. They don't allow the fore and hind wings to kind of gather together in flight. Doesn't allow the drones to fly as far, which has, repercussions in terms of mating potential. So that's one of the things that we talk about in terms of genetic situations in which genetic bottlenecks can create low queen quality. Then we have situations of poor mating conditions, and those can be a myriad, and I'm just going to bring up a few. Some of those is that the queen needs enough numbers of drones to mate with, and so in the spring, when the queen is not potentially having enough drones that are sexually mature early in the spring, she will be undermated. So you have to pay attention to the queen's natural mating conditions, especially early in the spring and late in the fall, when the availability of sexually mature drones and good quality drones might be low. So then you have to plan ahead and act accordingly. You need to ask yourself, even if you're just a beginner beekeeper or one that has very few colonies, you want to be cognizant of the mating conditions of your queens. Are there enough drones in the area during spring, early spring, or during the split time when you need queens to be the most vigorous so that they can lay a lot of eggs when you after you split a colony and you requeen it with a new queen? And in the fall, during queen replacement, if you're doing any kind of prophylactic or directed queen replacement prior to the winter to improve the colony survival through the winter and then improvement of queen performance in the following spring. And then, so you want to know what the quality or the genetic lineage or breed of the drone sources are nearby. In a study that we did here in my lab with my former PhD student, Adrian Fisher, who's now a faculty member at Arizona State University, we looked at the effects of coating, having wax in which drones are reared contaminated with miticides that we as beekeepers use to control for the varroa mite. And so we had comb that was miticide free, and then we had comb that was contaminated with the most common miticide that we use nowadays, which is amitraz. And we allowed those drones, the queens to lay in those frames, and then the drones to emerge, and we aged them with a paint on their thorax. And once they came out and became sexually mature, we then collected them and did the analysis of sperm viability. That is the percentage of the sperm in a drone or in the drone's semen that is viable and could inseminate a queen or be used up by a queen to fertilize eggs. And we found that contamination of comb with field relevant concentrations of amitraz significantly decreased the sperm viability of these sexually mature drones. So the queens may be mating with a sufficient number of drones, but those drones may be compromised in terms of the quality of the sperm because of the rearing conditions that they were reared in. And therefore, the queen kind of suffers from mating with standard drones. We published that paper a few years ago, titled "Exposure to Pesticides During Development Negatively Affects Honeybee Drone Sperm Viability," and that's open access, so anyone can actually read that paper. So that's one example. I basically, because we don't have a whole lot of time today, I can't talk about every study, but I'm just giving you some examples of each one of these factors. The other one is the age and the vigor of the queen. So you need to keep track of your queen's age. You nowadays with our smartphones, it's quite simple to take good quality photos of queens to know what that queen looks like in each of your colonies, unless you're, like a sideliner or a commercial beekeeper, and that becomes, it gets out of hand. But if you are a backyard beekeeper, which probably a lot of you are, then it's relatively simple to keep good records of your colonies and your queens. So you want to keep track of the queen's age, because if we don't know how old she is, then we're going to lose out on the queen's quality. So one of the things is that okay, give me 1 second because my husband is trying to get a hold of me. So give me 1 second because he's not here. Yes, Sammy, okay? Okay, so one of the things that can happen is that as the queen ages, the quality of the sperm that she's storing in the spermatheca goes down. And there was a study, a really good study that I really like, by Alawadi, that looked at the number of sperm cells in the spermatheca of queens that were mated over time. So this is a very famous photo by Sue Covey that shows that the queen, this is a spermatheca of virgin queen. Both of these spermathecae all spermathecae are covered by a tracheal net that keeps that spermatheca oxygenated and the sperm alive for the remainder of her life after she mates. So these two look like that after they've taken out the tracheal net. And this is the spermatheca from a mated queen. It looks marbly, kind of cafe au lait color, full of sperm. Generally, a queen that's been properly mated has about anywhere from 3 to 5 million sperm cells in her spermatheca, right as soon as she mates, which happens about 7 to 10 days after she emerges as a virgin. And so what they did with this study is they grabbed a large cohort of queens, all came from the same graft, same genetic background. They took one third of these queens, waited for them to mate, and once they mated, the sperm number analysis for 1/3 of them, and they calculated about 4 million sperm cells. Then the rest of the queens were allowed to go on with their lives for a year or so, and then half of the remaining queens, they looked at their sperm viability again. Sorry, in this case, sperm number, and they had already run out of more than half of the sperm just within a year. And then the remainder of the queens, they let live for another year, and then they did sperm viability a year later and sperm count, and they noticed that about half of those sperm were gone. So basically, within 2 years, the queens went from about 4 million sperm cells in the spermatheca to about only less than 1 million. So this shows how quickly a queen runs out of sperm. And this is under normal circumstances because it takes about 3 to 5 sperm cells for each egg to be fertilized. So she uses up about 3 to 5 sperm cells, one of which is successful in fertilizing an egg. So you can imagine she runs out of sperm really quickly. And so this decay or use of all the sperm is one of the reasons why we are having to replace these queens within a couple of years, because they basically run out of sperm prematurely. Another really cool thing that they did for that study in terms of the age of the queens is the motility of the sperm. And this has to do with oxidative stress, with how old the sperm is, how vigorous the sperm is, which will be used up later as the sperm comes down from the spermatheca, down the oviduct to fertilize an egg. So for that same study, what they did is they looked at the motility of sperm cells of really newly mated queens, and then 1 or 2 year old queens, the same ones that I just showed. So before they sacrificed them and looked at sperm number, they actually looked at the behavior of these sperm cells in the spermatheca. So newly mated queens, honeybee sperm has a really long tail and a very thin head, to the point that you can't actually see the head here in these photos. And they move, as soon as the queen mates, they move in circles, in concentric circles. And they look like spaghetti, like noodles, like ramen noodles that are kind of coiled up, and they move really fast and in circles. And they coil up with each other. And that's, as you can see in these cohorts, that could be one of the reasons why sometimes you have these little patches of the same parent having the offspring in kind of like patches. It's probably not that the queen is selecting which sperm to use, but these kind of noodle looking sperm kind of all go down the oviduct at the same time. But what they found is that over time, the sperm moves more slowly, and it changes the pattern of movement so that when now they have a lot more space, they move sideways. They move like in waves and they get elongated. They're no longer coiled up like this and they move slower. So that doesn't help with the migration of these sperm cells down the oviducts for the fertilization phase. So age, oxidative stress, and all of these things that come with getting old, especially in queens, has a big effect on queen quality. In terms of age as well, as I said, you need to keep track of your queen's age so you know what to expect, especially if you learned anything today about sperm storage in the spermatheca. So is your queen labeled? Do you have good records of your queen? Do you know when she was born, when you installed her in the colony? Have you labeled her? Even if you're not using the international nomenclature coloring code of the 5 numbers, any color will do. Some people might pinch with a scissors just a tiny tip of the fore wing, especially if she's of a very special kind, maybe an inseminated queen of sorts, just in case that the color code or the number tag comes off, you still have the same queen. But there's really kind of no excuses because nowadays we have our smartphones, so we could be using just photos, even if it's just photos of the queen, so we know that, you know, you can see the difference between these two. This one has the orange bands. This one is completely dark, so you don't get any surprises as to whether your queen was replaced by the workers in between inspections, and so there was supersedure and you didn't even know that you actually had a queen replacement scenario. So again, keep track of your queen's age, and always try to keep a vigorous bond. The reason why you want to know that you have a vigorous queen or that you don't is because then you might start seeing these signs by the workers doing what they know how to do, which is replacing an old or failing queen with a new one. And this is different than swarming, during which the mother queen leaves with about 3/4 of the worker population to found a new nest. In this particular scenario, if you have a failing queen, a queen that's old, ran out of sperm or is running out of sperm, or is ill, or just basically old and her pheromone cues are giving off the wrong message, then the workers will start trying to rear a queen from the middle of the brood nest area, and rear, instead of workers, a queen. So you probably know, female eggs are bipotent. They can turn into queens or workers based on the nutrition that the larvae get during the first 3 days of larval life. And so if the workers sense that the queen is failing, then they may start feeding any of these young larvae a diet of royal jelly that changes their developmental pathway into one of a queen. And so you start seeing these signs of queen cells in the middle of the brood nest area, compared to what you would find in swarming scenarios, which would be here at the bottom of cones. And so supersedure is a very telling sign, or at least supersedure attempts are a good telling sign that your colony is sensing that the queen is failing and it's time for her to go. And one of the reasons why is because she smells very strange. So the queen releases several chemicals from several glands around her body that basically keeps that decentralized control of colony maintenance, whereby she is the reproductive female and the workers perform all the non-reproductive tasks. So a queen-right colony functions as a cohesive unit, largely in part because of, among all of these chemicals that I told you, in particular, the queen mandibular pheromones, which are released by the mandibular gland and are transmitted around the colony through queen and worker contact. It's a blend of at least 5 different chemicals, and they're really long fatty acids and esters and all kinds of chemicals. And so their names are just shortened acronyms: 9-ODA, non-HDA, HOB, HVA. Here, non-HDA has a mirror image of plus and minus, and so that's what makes it 5. The reason why they found that it was these 5 in particular is because back in the day when they were trying to determine which chemicals these were, and they were isolating all of the chemicals one at a time, then they would create synthetic versions of these chemicals and see if there was a behavior of nurse bees taking care of the queen just by giving them these artificial chemicals. And that's how they decided that it was a blend of, in particular, 5 chemicals that comprise QMP. So these chemicals are produced by all over the queen's body. Imagine that these little dots are queen produced pheromones, but actually I think I will show later that it's not just QMP or the foregland or Turgel gland pheromones, but other chemicals. In any case, the queens that take care of the queen are called the retinue workers. And so they groom the queen, and as they do that, and while they feed her, they're also licking her, picking up her pheromones. And so each worker will either do self grooming, in which case some of these pheromones are translocated internally by that worker, which causes these behavioral and physiological changes, or they're going to do trophallaxis, food sharing, and they're going to pass down those molecules of QMP and other queen produced pheromones to the next layer of workers, such that every layer of workers around the colony, kind of in concentric circles around where the queen is, will receive a titer of queen produced pheromones that tells everyone she is there and she's doing well. So my former student, which I mentioned before, Liz Walsh, who is now at the USDA, did a few studies about the effects of these agrochemicals in wax on the reproductive quality of queens. And she did beautiful studies on this precious gland. So this is the head of a queen that's been decapitated, and the head has been placed kind of facing the bottom of a dissecting plate, and then she, with a scalpel, opened up the head cavity and took out these little drop looking glands that you can then extract the chemicals from. If you do that, if you extract the inside of one of those glands, you will find that QMP does many things, and I can't go over the whole list today. One of them is that it elicits this very fast or releaser response, which is a retinue response. So as soon as the workers smell that QMP, they will gather around the queen and start licking her, just like the diagram before showed, and tend to her, and then pick up those pheromones. So if you excise those glands and you put them on filter papers and you give it or offer it to nurse-aged bees, which are the ones that take care of the queen, so they have to be between the ages of 3 and 10 days post-emergence, they will be highly attracted to the contents of the QMP. So QMP elicits a retinue response. It also inhibits worker ovary development. So you may have heard that sometimes if you have a queenless colony, you can have rogue workers that turn into laying workers to lay unfertilized eggs, because they can activate a few ovaries that allow them to lay some unfertilized eggs. So this is a real photo of a laying worker ovary compared to a virgin queen compared to a mated queen. So their ovaries are never going to be as big as that of a queen, and of course they can't mate, so they can't fertilize those eggs. But if you see something like this with multiple eggs per cell, that's a telling sign that your queen is gone, and these workers have, because there's no QMP anymore, it kind of, that inhibition is lost, and some workers have developed their ovaries and started to lay unfertilized eggs, which is actually difficult to overcome if it's been too long since the colony has been queenless. There was a paper that now is not that recent, but was published in 2019, that I love because it kind of opened our eyes to a new set of queen-produced chemicals that hadn't been thought of before. So every time we read about papers, it was QMP, maybe the fourth gland, Turgor gland, egg marking pheromones. But this one is titled Honeybees Possess a Structurally Diverse and Functionally Redundant Set of Queen Pheromones, which basically the punchline is that they discovered that they also produce cuticular hydrocarbons. So they're produced by the exoskeleton and the Turgor glands. And so not just the QMP is what maintains this inhibition of worker ovary activation, but other chemicals. So what they did was they dequeened a bunch of small colonies. They let them be queenless for a little bit so that the workers sensed that queenlessness, and then they started activating their ovaries, and those were the control colonies. But in some of them, once they had dequeened the colonies, they then added these synthetic chemicals like 9-ODA on its own, 9-HDA on its own, HOB, HVA, or the combination, which is the 5-chemical blend. And there was a decrease in the amount of worker ovary activation, or the number of workers with developed ovaries, which is not surprising because we've known this for 30 years now. But what was cool about that study is that again, they found these novel honeybee pheromones, including cuticular esters, cuticular alkenes, Turgor gland esters, and Turgor gland acids that did the same thing, reduce the number of workers with developed ovaries. And therefore, as this says in the title, redundant set of queen pheromones that help prevent workers from developing their ovaries. And so that basically is one of the other things that I wanted to mention about mating conditions of the queens and how old they are, because depending on how old they are and whether the queen is ready to be replaced, then the production of all of these chemicals is going to be different, and therefore the workers are going to detect those differences and act accordingly. Okay, so pesticides have a lot to do with our maintenance or management of queens. We have to be cognizant of all kinds of chemicals that may harm the colony, and in particular with respect to queens. So I mentioned the study that my former PhD student Adrian did with drones. Well, we did a similar set of studies with Liz, but it was with queens. So what happens with you when you have comb that's contaminated with field relevant concentrations of what we as beekeepers apply to the colony to combat the varroa mite, miticides. And so we reared queens in cups, plastic cups that were coated with wax so that they looked like normal queen cups in the field. But those cups were contaminated with field relevant concentrations of combinations of these miticides or agrochemicals. Once those queens emerged from their cells, we allowed them to mate naturally. We did a few experiments with these queens, but one of them was to put those mated queens into 3-frame observation hives like this one. And we basically looked at the queen's behavior and the queen's treatment by the workers. So we had these observation hives that had anywhere from 5,000 to 6,000 workers. And then we measured a couple of things. I'll mention just 2 for this particular example. One was the retinue size, so the number of workers around the queen at any given time. So you can see this is a very robust queen retinue with about 12 workers. So she's very attractive. they love this queen and they want a lot of her pheromones. Whereas this is a very weak pheromone profile, a very weak retinue response with workers not really liking what they're smelling. So we measured that retinue size by counting the total number of workers that we counted per minute for 5 consecutive minutes per queen. And we had control queens, those that were reared in miticide-free wax or chemical-free wax, and those that were reared in chemical-laden wax. And we did at least 50 5-minute observations per queen for each treatment group. And then separately, we also followed the queen's egg laying rate, which is counting the total number of queen eggs that the queen laid per minute in a 5-minute interval. So what we found was that there were significantly fewer workers around the queen's retinue in those colonies that were reared, in those colonies whose queens were reared in wax that was contaminated with either amitraz, with the old miticides that we used to use in the 1990s, 2000s, but are still present in wax, fluvalinate and coumaphos. And then the two most common agrochemicals that were found in wax at the time, chlorothalonil and chlorpyrifos. So the number of bees in the retinue was significantly lower in all of those contaminated wax queens, which has direct implications because that means fewer bees taking care of the queen, less feeding, less QMP, and all of these chemicals being passed around the colony. And what's even more striking was the number of eggs. So the egg laying in a 5-minute interval. So the same thing. Control queens laid significantly more eggs than any of the two miticide groups, not for the other agrochemical group. There were no differences. So if you see like 0.5 eggs, of course, that makes no sense. 0.5 eggs in 5 minutes, it's not a number that we can think about. But imagine that the queen, if we imagine that these queens are laying the same rate of eggs throughout a year, which is not really true because they will stop laying eggs in the winter. But if they kept this egg laying pattern for a year, by extrapolation, these control queens would lay about 200,000 eggs per year. And comparatively, the ones that were reared in fluvalinate and coumaphos laden wax would lay about 132,000 eggs, which is a decrease of 35% compared to the control. And even more striking, those that were reared in amitraz laden wax would lay only 115,000 eggs, which is a decrease of 43% compared to the control. And this is the miticide of choice nowadays in the US. So you can see how something as simple as having wax that's contaminated with these field relevant concentrations of miticides can have severe downstream effects in terms of colony productivity, number of eggs. Something really interesting that we also found was that we looked at the number of mates that the queen mated with. And those that mated, especially with this group, those that were reared in the fluvalinate coumaphos laden wax, had an abnormally high mating frequency compared to the controls queens. And what's interesting is that I had actually found the same effect in a previous study that I had done when I was at North Carolina State. And we still don't know what this is telling us, but we basically found the same results that these queens reared in miticide-laden wax. Maybe something about the stretch receptors that tells them that they've been fully mated is not working, and so they end up overcompensating by mating with too many drones, which might not be necessarily a good thing. We don't know exactly how this works, but this was an interesting result. So as I said, this was also published before. So the quality of the queens can be affected by exposure to pesticides inside the hive. Now, we know that the worst parasite that we've encountered so far in apiculture in the last 50 years is Varroa destructor, but there's also other pathogens that queens get exposed to. So you can actually have either queens or drones that have high levels of Varroa or Varrosis. And so when your colonies have really high Varroa levels, you can actually have queens that also get Varroa during development. And so I've found a lot of, well, not a lot, but a few queens um, that we're taking out of sales and they actually have Varroa on them. Um, and as you probably know, Varroa destructor vectors several honeybee associated viruses. In particular for queens, one is the form wing. So this is a picture of a queen that was heavily infected with the form wing virus during pupation. And so she emerged with the telling symptom of shriveled wings, which basically tells us, of course, that she will be unable to mate, and she probably wouldn't have lasted very long because she would have gotten probably superseded immediately. And then they also get things like black queen cell virus, in which case they will never emerge as adults because they die in the pupal state inside the cell. Another thing that they can get, and it's actually something that we're currently studying in our lab, is nosemosis. So the disease that's caused by either Nosema apis or Nosema cerani, the microsporidian gut pathogens. Nosema apis causes the telling sign of dysentery or diarrhea because the bees basically can't go and void themselves outside of the hive like they normally would. If they're heavily infected, they must have several million spores in their gut for them to be severely infected. Nosema cerana infection is not as symptomatic. Um, and there's actually been very few studies that have looked at the effects of Nosema cerana infection on queens. One was done about 15 years ago, looking at what nosemosis does to queens. And they found that queens that were infected with Nosema cerana in particular um, had significantly, not significantly, they had numerically, but not significantly lower fat body tissues, which, if you know, fat bodies help produce vitellogenin, and it's a good sign of being healthy and having a lot of food reserves. They had higher vitellogenin levels, significantly higher vitellogenin levels, which when you have higher vitellogenin levels, it's an indication that the colony and also antioxidative capacity, they're trying to combat infection. And so Nosema cerana infected queens had higher markers of infection issues. But what's interesting to me is that they also cause significant changes in the production of QMP chemicals. So Nosema cerana infected queens had significantly higher levels of 9-ODA, of 9-HDA, and significantly lower levels of HOB, which means basically that the whole pheromone bouquet smells very differently and poorly to workers, probably poorly to workers, because these queens smell off. So what we're doing in my lab right now, which we've actually found, but we haven't published this, is that we took those queens, infected them with nosema, and we looked at the other things that we've looked in the past at retinue behavior, egg laying capacity, motility or mobility, and we found that all of those behaviors are altered by nosema infection. So we'll probably be publishing all of those results next year. I'm almost done. I just have a couple more, few more slides. One is nutrition. So the colony needs to have enough good nutrition for not just the workers, but also the queens to have, ideal health conditions. So a few years ago, there was a study that looked at these microcolonies, how to rear small colonies in the lab that allows us to watch queen behavior properly And this was Julia Fine when she was at Gene Robinson's lab. So she tested the efficacy of these three different um, um, models of these little mini cages where she had all these feeders and allowed the queens in there with a few workers to lay eggs in those cells. And so the workers would be the ones that feed off of all of these feeders. You give them specific diets, then those workers take care of the queen, and then you see what the queen's behavior is like. So they followed the queen when they fed those microcolonies either normal bee bread or BB, which is the equivalent of the pollen when it's stored in the hive, frozen bee bread, or a pollen patty. And so the workers in those microcolonies ate more of the pollen patty, but the queens, when they were laying eggs, laid more eggs per unit time over time when they were fed the real deal bee bread, either frozen or unfrozen bee bread, compared to the pollen patty, which tells us that um, queen nutrition is compromised to some degree, at least in these microcolonies, right? When the bees are fed an artificial diet compared to when they get fed normal, well, in this case, brood food or food from the workers, when the workers have access to fresh bee bread compared to pollen substitutes. We know that we have issues with habitat fragmentation, urbanization, destruction of habitats. And so urbanization causes the loss of suitable habitats, and this it's no different for queens, right? So if the colony doesn't have access to forage, then the workers cannot feed the queen and drones proper amounts of food. Climate change, either extreme cold or extreme drought, are both bad because you can change the phenology of pollinated plants, causing either early plant flowering or causing things to run out of not having enough nectar or pollen because of a drought, or extreme storms can wash off all the nectar and pollen and not allowing the bees to feed, which then causes malnutrition either way in the colony. In terms of temperature, which is probably what's happened, like right now here in Texas, this week has been record high temperatures of in the mid 90s, which was unbearable. This is probably our new normal, is these erratic temperature fluctuations. There was a study actually many years ago, it's in 1994 by Marla Spivak, that was kind of testing this idea of what happens when you rear queens in above ideal temperatures during development. And so she tested 3 different temperatures during pupation of a black and a yellow line of bees. And she noticed that as the temperature went up during development, the developmental time of the queens went down. So instead of 18 days, it went down to 15 days. So a difference of 3 days. So for every like 2 degrees, there's a decrease of about a day in development, which is not ideal because that means that those bees are probably not getting enough food during the larval stage, which I bet you, even though they didn't look at this, the bees were smaller in size when they emerged sooner than normal. And then finally, these beekeeping practices. So having poorly mated queens or very old queens that cause supersedure events. Again, the drone laying queens that don't ran out of sperm, and so they're become drone laying queens. So if you're looking at a completely all worker drawn comb, and you have the situation where you see a lot of dome-shaped large cells that look like drone cells, but they're in worker cells, that means that your queen started laying unfertilized eggs, and so the workers will raise them as such, as drones. And so this is a telling sign that you have a drone laying queen, which of course needs to be replaced. If you see these telling signs of a lot of cells in the middle of the brood nest area with these exit holes, you probably have a new queen. Your colony got either an emergency queen rearing situation where the queen had already died, or they replaced her because she was too old or smelled weird or she ran out of sperm. Or if you see this, as I previously mentioned, then you probably have been queenless for a while, or your colony, and then you have this hopelessly queenless situation where the workers have laid several eggs per cell. If your colony is hopelessly queenless and you have a bunch of drones that are produced by these laying workers, some people might ask, what happens? Well, if your queen is gone, well, you might as well get the genes from the drones, from those workers, right? Well, there was a study that looked exactly at the question of whether the sperm traits of drones from laying worker colonies is the same or different from queen-right colonies. So they looked at the drones from the queen-right colonies and drones from laying worker colonies. And for every trait that they looked at, weight, volume of the ejaculate, the number of sperm cells, the concentration of sperm per unit volume, and the sperm number for milligram of body mass, they were all significantly lower in the laying worker colonies. So they're smaller drones that are probably not as efficient as those from queen-right colonies to mate. So this is all kind of a compendium of things of queen management essentials. It's advantageous for us nowadays because of all these issues with poor queen quality to requeen your colonies every 1 to 2 years, because a young productive queen can translate into populous colonies that have either higher honey production or have a lower tendency to either abscond or collapse. Those colonies are headed by first-year queens, as I just said, are less prone to swarming. Failed requeening attempts will halt colony buildup. Requeening as in natural requeening by the colony may halt colony buildup. A colony's own requeening attempts, if they fail, they can endanger honey flow or overwintering. And because of all of what I said previously, the longer that a colony is queenless, the harder it is to requeen later on, especially because you have that potential for workers to start laying eggs. And there's this disarray because there's not the proper combination of queen-produced pheromones in the colony. So I hope that I gave you basically a few tips. If you got at least one that you didn't know before in terms of queen management essentials, I would feel satisfied. There's so many factors, and of course I could talk for another hour on all of these nuances and even more research that's come out in the last 5 to 8 years in queen health that we didn't know before. But these are kind of the main ones that you need to be on the lookout for. And so we're always playing detective, basically, and trying to understand what's going on with our colonies and our queens. But the ideal scenario is that you don't have to do a post-mortem assessment, but that you can catch these problems before it's probably too late and your colony can suffer from basically overwintering mortality. So I don't know if I have any time for questions, but here's our very popular Facebook page, TAMU Honeybee Lab, where we post all of our most up-to-date activities at the lab. We just had on Saturday and Sunday our queen rearing workshop with Sue Kobe as the person that does instrumental insemination demonstrations, and Megan Mahoney from South Texas, which is a commercial queen producer, and then a bunch of other people that are either faculty or commercial queen producers in our state that love to teach about how to rear good quality queens in our state.
Thank
So
you very much.
I'll
Thank you
be
very
happy
much,
to take
Juliana.
any questions if we have time.
Thank you. Yeah, and we have time on our end. There are a couple of questions in the chat. I don't know if you can see those ones. Pete put a few questions in the chat. So any of
Oh,
the if you would like to answer any of those.
okay. So if the queen pheromones block ovary production, wouldn't this indicate that the queen's bred a supersedure would be less successful if you raise a queen as part of a simple split? To indicate that the queen raised as a supersedure. Well, yes, so there's the chance that a colony, when you're when the you leave it up to the workers to do the supersedure, our studies and others have shown that there's this flexibility in the product in kind of a grace period during which a queen can be produced. They can feed worker larva royal jelly immediately after the egg hatches, or well into the first 24 hours, 48 hours, 72 hours. So for the first 3 larval instars. The longer that the workers wait to feed that larva royal jelly, the smaller the queen once she emerges as an adult is going to be, and the less productive that queen is going to be as a mom. She's going to produce fewer eggs, and her population is going to be smaller. The food production for that colony is going to be lower, and all of these metrics decrease by 20 to 40%. So yes, because sometimes when workers supersede, they only have access to older larvae, or they choose to rear a queen from an older larva, and we still don't understand how they make that decision, but they do it. And so if they chose a 2 instar or 2-day old larva instead of a 1-day old, then that queen is not going to be as successful later on.
Thanks, you answered. That's perfect. Thank you.
Yeah.
I didn't mean to hog all the questions. I'm happy to do it.
As a small time beekeeper, what about with 16 hives in one location? Would you be in trouble from inbreeding if you replace all your queens with ones that were all grafted from the same frame of larvae? They would be open mated in boxes next to the apiary. So I don't think so. So this is the one of the issues with having a small operation, is that you can't really control the mating of your queens. All you can do is hope that there will be enough diversity in your vicinity, because your queens are all going to go and mate over a mile or kilometer away from where your hives are. And so they're not going to be inbreeding, because they're not going to be mating with drones from those 16 colonies. Drones tend to fly only a couple of 100 yards away at most from their home colony. And that's a very important, super important evolutionary strategy for Apis mellifera to avoid this inbreeding problem of producing diploid drones. So what's going to avoid this inbreeding likelihood is the fact that the drones in a kind of a kilometer and a half or so away from where your colonies are not related to your queens.
Right. So that would indicate there's a drone collection area within a couple hundred yards of my apiary. Is that what
There's
you're saying?
no, what I'm saying is that your queens are going to outbreed.
I understand that. I'm just saying that I've also got a whole bunch of drones coming out of those hives, and they go somewhere. So I would assume they're going to a drone collection area, and it would probably be within a couple hundred yards of my hives.
Yeah, but your queens are not going
I
to.
know they're not mating there. It's just a separate question.
Yeah, so yeah, I heard from because we just had the workshop Saturday and Sunday. Sue was saying that there's a study that showed that queens will actually fly as soon as they leave the hive. They go at a completely different height than the drones to avoid the drones that are from the nearby area, and then they kind of overshot anything that is a potential cloud of drones nearby so that they can go and to somewhere else. So the inbreeding would come more from them breeding with related drones, which is not very.
Not likely. Yeah, that's fine. And then the last question I have.
The inbreeding problem would come if, for any reason, you had, and this is for bigger scale beekeepers, is if you had drone source colonies that are related to your queen producing colony that are a mile or so away in the area where the queens go mate, then that would increase your chances of inbreeding.
Right. Thank you. The last question I was just, I thought Amatraz broke down. So are you putting?
So what we did, it was it's the metabolite of Amitraz.
So it's the part that it breaks down to some formaldehyde or
MPF or something like that.
Yeah,
the one that we tested. Yes, that's right.
Okay, that answers probably the whole thing then, because that does show up in the wax then? Is that what you're saying?
Absolutely.
Okay, so when somebody says Amatraz doesn't show up, it's the product doesn't show up, but the metabolite does.
Yes, it does at high concentrations. But what's very strange in that study that we based these studies off of, that one was in 2010, which is now dated, but then they have done similar studies looking at wax pesticide residues in 2016 and 2019, and they still found fluvalinate and coumaphos in wax. And we haven't used those supposedly, since 2000, and it's still prevalent in wax, which might tell us it's either being picked up from other agricultural production practices and being brought in, or because of the high concentrations. I mean, they're the top two chemicals found in wax still. The recycling of wax, and that we're not cleaning the wax well enough when we buy wax commercially, like foundation and other types of wax.
Interesting.
Remnant of these chemicals. That wax is highly lipophilic, and all of these chemicals acts like a sponge. Yeah.
So even if you use it for just like 1 month or 2 months of the year, because we don't need it for very much, and then use formic acid or oxalic acid afterwards, you're still getting this buildup in your wax, huh?
Yes. However, I should say a disclaimer, and I should have mentioned this. I'm trying normally I have a whole talk just for those studies with pesticides. These were field relevant concentrations, but mostly for commercial operations. So they're high compared to what you would find in wax of, let's say, backyard beekeepers that don't have such an intense use of miticides to keep the mites away. So this is probably the worst case scenario of heavily contaminated wax. Having said that, though, just a one-time application of these chemicals in the wax, and it was just on the coating of the bottom of the cells for all these experiments, was enough to see an effect.
Interesting.
So they probably the chemicals leach onto the food that's being fed to the queens and the drones. That's probably the way that it gets to them.
Makes sense. Thank you.
I have a quick question about the timing of requeening, because you mentioned the importance of potentially requeening on a kind of regular basis to make sure that our queens are not old, and then also the idea of timing that and making sure that our colonies are not queenless for a longer period of time, because then it's more difficult. But on the shorter end of that time, is there an ideal time between, let's say, killing a queen and introducing either a cell or a mated queen? Is there an ideal kind of best practice with that? Is it important to leave a certain amount of time before introducing that cell?
Oh, yes, absolutely. So you want to kind of wait until the QMP of the former queen dissipates. So you want to probably leave the colony queenless if if it's not queenless already, for maybe at least 1 day, if not 2. And then when you introduce the new queen, there are several techniques. One is to spray the cage that she's in with some sugar water, spray some of the bees around where you put the cage with some sugar water so that they're busy just licking and then they get to like the queen. And definitely either use candy or a plug and then let her be in that cage for at least a day or 2. And importantly, look at the behavior toward the queen as soon as, if you're doing cork release, right? If you are going back to release her, then see how the workers treat her immediately. Even if before that, because with the candy, she may emerge, that the idea is that you don't have to go back. So you may have to go and find her. But when you're doing the one where you have to take out the cork and release her, the cap, even before that, you will see how the workers behave through the mesh. Queens that are not liked by the workers, even after 1 or 2 days, you're going to have a clear sign of aggression of workers trying to bite through the mesh and basically trying to harm her because they want to get rid of her. That would be like trying to do a killing of a queen. And they do that. When they do that, they can harm her depending on the size of the cage, to the point that they chew off her tarsal claws, and they take off the little hooks that they have at the end of the feet, so that they may emerge and they may actually be ultimately accepted, but they're already harmed, they're injured, and that can lead to super seizure because they've already been injured before they even had a chance. So there's a lot of different kind of techniques that you can do to lower the chance of failure in an introduction of a queen. It's very tricky because there's potential problems at every step of the way. Also with queen rearing, because we were just doing the queen rearing workshop, and you lose queens along every step of the way, during grafting, during the percent take in the cell builder, how many cells actually got capped, how many queens emerge from their cells, how many queens mate, of those how many get accepted. So you can kind of understand then sometimes why queens are so expensive to purchase, especially if they're guaranteed mated queens, is because there's so many things that can go wrong with queen reintroduction.
I have a question. Would it be wise to get rid of your old wax and let them draw out new foundation every year?
Not every year, not for someone that does some backyard operation. I would probably do something like every 4 or 5 years. And one good telling sign is the darkness of your comb. If you have really, really dark comb, as it gets older, it gets darker, then something that looks black is probably not very new. And so you probably have a bunch of chemicals there. But depending on how many colonies you have, it might get kind of expensive. So you might want to do a rotation system where you only replace maybe 10% of your frames each year or something like that to lower your costs. But I wouldn't do it every year. Definitely if you're a backyard beekeeper, unless you suspect that there's a lot of agricultural chemicals, for example, around you. And it also depends on how much miticide use you have in your operation.
Okay, thank you very much.
Do you see any issues with oxalic acid or formic acid in terms of this kind of thing, or is it?
That's a great question that I get asked all the time. So all this work was done a few years ago when we had money for that. At the time, I was, I proposed to do studies on looking at these organic acids. But because they don't stay in the wax,
Mhm.
it wasn't something that got any traction. But I have heard of anecdotal accounts of queens failing after a heavy dose of OA, for example. So I'd love to do a study on that, but I haven't pursued it yet, and I haven't seen anything yet. I suspect that there has to be some sublethal effect on the queens, depending on when it's applied and
Yeah.
quantity, but to my knowledge, no one has answered that question yet.
I know formic acid can kill a queen pretty quickly if but it seems very random. I've never been able to see exactly why. I tried to kill a queen with formic acid in a hot hive, and I went in 3 weeks later. The hive was bagged, and I had like 3 pads of formic pro in there. Most of the bees were dead, but the queen was walking around. So I was like, how can you not kill a queen?
How are you not dead? Yeah,
Yeah.
it's
So I don't know, but then I do it.
really fragile. So it's
Yeah.
yeah. But stay tuned. Maybe, in a couple of years, I'll get some money to look at that in particular.
Well, thank you once again. Thank you for coming to present to us. We loved your presentation, and thank you for taking the time to answer some.
Thank you. I'm
Well,
sorry that I couldn't find the right Zoom link. I apologize.
no problem at all. I'll work with our other people on the board to send out an announcement about the event on Tuesday, that education event. So we'll get that out to our club members as well.
I can send you the flyer if you want.
Perfect. Thank you very much. And I'll pass that along to our club members. And once again, thank you very much. For those of you joining us tonight, thank you. Yep, thank you.
Thank you.
We'll be having another Zoom speaker series in June, so stay tuned for that. That'll be in the newsletter. And thank you, Professor, once again for joining us.
You're welcome. Thank you so much. Take care.
Here you go.
Bye.
Bye.
Bye. Dan. Bye.